Good evening, everyone. So today we'll start our 10th live session of this course RNA Biology. So as you know, in these live sessions what we do is that we discuss some previous year assignment questions, and when we discuss those solutions, we not only discuss the solution to that particular problem, but we also discuss similar type of other questions, so that it's easier for us, we understand the concept behind this question, so it's easier for us to understand the topic properly and to solve similar type of other questions. Okay, so we'll start today's session, and we tend to keep these sessions as interactive as possible so that it helps us and it helps you. Okay, so if you have any questions, if you have any doubts, if you know the answer to any questions, please write it in the chat box or unmute and let me know, and we can take it up from that. Okay, so moving on to the first question for today. Bacterial mRNA codons are A. Not similar to plant codons, B. Similar to other animal codons, C. Not similar to virus codons, D. Similar to mitochondrial codons. So I'll wait for a few minutes and I'll let you come up with the answer, and then we'll discuss the question. Anyone? Okay, so the answer would be similar to other animal codons. Okay, so what happens in mRNA or messenger RNA? Codons are groups of 3 consecutive nucleotides that specify a particular amino acid during protein synthesis, forming the genetic code. So what are codons? Codons are triplets of nucleotides A, U, G, C, adenine, uracil, cytosine and guanine, right? That are read sequentially along an mRNA molecule. Now each codon corresponds to a specific amino acid or in some cases signals the start or stop of translation. Now the relationship between mRNA codons and amino acids is called the genetic code. And this code is nearly universal, meaning that the same codons specify the same amino acids in most organisms. Now if we talk about start and stop codons, the start codon AUG signals the beginning of translation and codes for the amino acid methionine. And the stop codons UAA, UAG and UGA signal the end of translation and do not code for any amino acids. Now during translation, mRNA codons are read by transfer RNA molecules which carry specific amino acids. The tRNA molecules recognize the codons on the mRNA and deliver the corresponding amino acids to the ribosome where the polypeptide chain is assembled. Now if we talk about the mRNA structure, it's a single-stranded RNA molecule that carries the genetic information from DNA to the ribosome. And the mRNA sequence is composed of 4 nucleotides, adenine A, guanine G, cytosine C and uracil U. We know about the genetic code. Now if we talk about the overview of translation, we have discussed about translation in many of the previous classes. Now the second stage in gene expression is translating the nucleotide sequence of a messenger RNA molecule into the amino acid sequence of a protein. Now the genetic code is defined as a relationship between the sequence of nucleotides in DNA or its RNA transcripts and the sequence of amino acids in a protein. Now each amino acid is specified by one or more nucleotide triplets, codons, in the DNA. Now during translation, mRNA acts as a working copy of the gene in which the codons for each amino acid in the protein have been transcribed from DNA to mRNA. Now tRNAs serve as adapter molecules that couple the codons in mRNA with the amino acids they each specify, thus aligning them in the appropriate sequence before peptide bond formation. And translation takes place on ribosomes, complexes of protein, and rRNA that serve as the molecular machines, coordinating the interactions between mRNA, tRNA, the enzymes, and the protein factors required for protein synthesis. Now, many proteins undergo post-translational modifications as they prepare to assume their ultimate roles in the cell. Now, if we talk about the genetic code, most genetic code tables designate the codons for amino acids as mRNA sequences. The important features of the genetic code include each codon consists of 3 bases or triplet. There are 64 codons. They are all written in the 5' to 3' direction. 61 codons code for amino acids. The other 3, UAA, UG and UAG, are the stop codons and the nonsense codons that terminate translation. And there is one start codon, initiation codon. AUG coding for methionine. Protein synthesis begins with methionine in eukaryotes and formylmethionine FMET in prokaryotes. Now the code is unambiguous. Each codon specifies no more than one amino acid. Now the code is degenerate. More than one codon can specify a single amino acid. All amino acids except methionine, and tryptophan have more than one codon. For those amino acids having more than one codon, the first two bases in the codon are usually the same, and the base in the third position often varies. So the code is almost universal, the same in all organisms. Some minor exceptions to this occur in mitochondria and some organisms. So the code is commaless or the contiguous, right? So there are no spacers or commas between codons on an mRNA, and neighboring codons on a message are non-overlapping. So this is the basic table of a genetic code. Okay. So if you see, this is how the genetic code table has been formed. Now, if you talk about the wobble hypothesis, I think I've already discussed it before in one of the classes, but let's talk about it again. Many amino acids are specified by more than one codon, which is what we call redundancy, right? Because there are more codon sequences that can specify one particular codon. Now, frequently a tRNA can translate more than one of these codons, sparing the cell from making multiple tRNAs to carry the same amino acid. For instance, the arginine tRNA arginine can translate both CG and CGG codons that specify arginine. This phenomenon is known as wobble and can be summarized like this. Correct base pairing is required at the first position of the codon, like third of anticodon, and the second position of the codon, second of anticodon. The third position of the codon does not always need to be paired with the anticodon. That is, it is allowed to wobble in some cases. Okay, so this is what we call the wobble position. Okay, if you see, this is the tRNA that will bind, okay, and the mRNA that goes for the protein. Now, if you talk about mutations, a mutation is any permanent heritable change in the DNA base sequence of an organism, and this altered DNA sequence can be reflected by changes in the base sequence of an mRNA and sometimes by changes in the amino acid sequence of a protein. So, mutations can cause genetic diseases. They can also cause changes in enzymatic activity, nutritional requirements, antibiotic susceptibility, morphology, antigenicity, and many other properties of cells. Now, transition is a point mutation that replaces a purine pyrimidine base pair with a different purine pyrimidine base pair. For example, an AT base pair becomes a GC base pair. A transversion is a point mutation that replaces a purine pyrimidine base pair with a pyrimidine purine base pair. For example, an AT base pair becomes a TA or CG base pair. Mutations are often classified according to the effect they have on the structure of the gene's protein product. So this change in protein structure can be predicted using the genetic code table in conjunction with the base sequence of DNA or mRNA. If we talk about some common types of mutations in protein structure, silent or new codon specifies same amino acid. The effect on protein is none. So that's why it's called silent mutation, because even though there is a mutation, the amino acid is not changed. So there is no effect on the protein. Then missense. New codon specifies different amino acids. So they can possibly decrease in function, and there can be various anticipatory effects. Then there's nonsense. New codon is a stop codon. So that's shorter than normal, usually non-functional. So if there's a new codon that basically equals for the stop codon, that will basically terminate your translation beforehand. Dark segment deletion, unequal crossover in meiosis that will lead to loss of function shorter than normal or entirely missing. Splice donor or acceptor, variable effects ranging from addition or deletion of a few amino acids to deletion of an entire exon. And triplet repeat expansion. Expansions in coding regions cause protein product to be longer than normal and unstable, and disease often shows anticipation in pedigree genesis. Okay? So these are the types of mutations. The types of mutations: normal, silent mutation, missense mutation, nonsense mutation, friendship mutation. Now, if we talk about protein synthesis, if we talk about the kinds of RNA, the class of RNA found in ribosomes is called ribosomal RNA. During polypeptide synthesis, rRNA provides a site where polypeptides are assembled. Transfer RNA molecules both transport the amino acids to the ribosome for use in building the polypeptides and position each amino acid at the correct place on the elongating polypeptide chain. Human cells contain about 45 different kinds of RNA molecules. Then we talk about messenger RNA. Molecules are long strands of RNA that are transcribed from DNA and that travel to the ribosomes to direct precisely which amino acids are assembled into polypeptides. Now, if we talk about the central dogma, information passes from the gene's DNA to an RNA copy of the gene, and the RNA copy directs the sequential assembly of a chain of amino acids. Then we talk about the genetic code, which we've already discussed. The essential question of a gene expression is how does the order of nucleotides in a DNA molecule encode the information that specifies the order of amino acids in a polypeptide. So the answer came in 1961 through an experiment led by Francis Crick. The experiment was so elegant and the results so critical to understanding the genetic code that we will describe this in detail. So proving code words have only 3 letters. So Crick and his colleagues reasoned the genetic code most likely consisted of a series of blocks of information called codons. They further hypothesized that the information within one codon was probably a sequence of 3 nucleotides, specifying a particular amino acid. So they arrived at the number 3 because a 2-nucleotide codon would not yield enough combinations to code for the 20 different amino acids, right? That commonly occur in proteins. So with 4 DNA nucleotides GCTA, guanine, cytosine, thymine and adenine, only 42 or 16 different pairs of nucleotides could be formed, right? Now, however, these same nucleotides can be arranged in 43 or 64 different combinations of 3 more than enough to code for the 20 amino acids. So when they made a single deletion or 2 deletions near each other, the reading frame of the genetic message shifted and the downstream gene was transcribed as nonsense. However, when they made 3 deletions, the correct reading frame was restored and the sequences downstream were transcribed correctly. So they obtained the same results when they made additions to the DNA consisting of 1, 2 or 3 nucleotides. So the discovery of the genetic code was a significant milestone in molecular biology and multiple scientists contributed to this field. So it would be wrong to only talk about Crick and not mention the other people, and by mentioning his colleagues, we are basically disrespecting their work. So if we talk from the beginning, so before the genetic code was deciphered, scientists had begun to speculate on how genetic information might be translated into proteins. So the first was George Gamow, a theoretical physicist. Gamow proposed the diamond code hypothesis, suggesting that groups of nucleotides encode amino acids. While incorrect, his ideas helped stimulate research in this direction. Then we have Francis Crick. Crick postulated the adapter hypothesis, suggesting that there must be molecules later which were identified as transferred RNAs, tRNAs, that help translate the genetic code into proteins. He also proposed the verbal hypothesis, explaining redundancy in the genetic code. But the experimental deciphering of the code was mostly done by Marshall Lindenberg and Johann Mattei. So at the NIH, Nirenberg and Mathai performed a key experiment using a synthetic RNA sequence composed of only uracil, like poly-U. So they discovered that it detected synthesis of a protein composed entirely of phenylalanine, proving that the triplet UUU codes for phenylalanine. Then we have Severo Ochoa and Marianne Grunberg Manago. They discovered polynucleotide phosphorylase, an enzyme that enables the synthesis of artificial RNA sequences, which help in displaying the genetic code. Then we have Hargobind Khurana. Khurana developed methods to synthesize defined sequences of RNA, allowing systematic identification of codon amino acid relationships. So his work established the assignments of many codons. Then we have Robert Hawley. Hawley sequenced the first tRNA molecule, confirming Crick's adapter hypothesis and showing how amino acids are linked to specific codons via tRNA. So Nirenberg, Khurana and Hawley. So the full genetic code was deciphered, showing that it consists of 64 codons, where 61 code for amino acids and 3 codons, UAA, UAG and UGA, function as stop codons. So in 1968, Marshall Nirenberg, Hargobind Khurana and Robert Hawley were awarded the Nobel Prize in Physiology or Medicine for their contributions to cracking genetic code. So basically, genetic code was not the result of one person or someone just whose work has been demeaned by mentioning as colleagues. Genetic code was deciphered through a combination of theoretical insights, biochemical experiments, and synthetic RNA technology. So the collaborative efforts of Nirenberg, Khurana, and Hawley and others played a crucial role in this discovery, which remains one of the greatest achievements in molecular biology. Right? So this is how it was done. If we delete one base, like for example T, it will lead to a nonsense RNA because hypothesis is unpunctuated. If you delete T, so this is if you delete 3 bases, that does not make some difference. So the code is practically universal. For example, the codon AGA specifies the amino acid arginine in bacteria. In humans and all other organisms, the genetic code has been studied. So that answers our question. Bacterial mRNA codons are similar to all other animal codons, right? Now, because the code is universal, genes transcribed from one organism can be translated in another. The mRNA is fully able to dictate a functionally active protein. Similarly, genes can be transferred from one organism to another and be successfully transcribed and translated in their new host. So many commercial products such as the insulin used to treat diabetes are now manufactured by placing human genes into bacteria, which then serve as tiny factories to turn out prodigious quantities of insulin. Okay, this is genetic code. In 1979, investigators began to determine the complete nucleotide sequences of the mitochondrial genomes in humans, cattle, and mice. It came as something of a shock when these investigators learned that the genetic code used by these mammalian mitochondria was not quite the same as the universal code that has become so familiar to biologists. So in the mitochondrial genomes, what should have been a stop codon UGA was indeed read as the amino acid tryptophan. So AUA was read as methionine rather than isoleucine, and AGA and AGG were read as stop rather than arginine. This it appears that the genetic code is not quite universal. Now, genes are first transcribed and then translated. The first step in gene expression is the production of an mRNA molecule, copy of the DNA sequence encoding a gene. The process is called transcription. And to understand the mechanism behind the transcription process, it is useful to focus first on RNA polymerase, the remarkable enzyme responsible for carrying it out. So RNA polymerase is understood in bacteria. So bacterial RNA polymerase is very large and complex, consisting of 5 subunits. 2 alpha subunits bind regulatory proteins, beta prime subunit binds the DNA template, beta subunit binds RNA nucleoside subunits, and sigma subunit recognizes the promoter initiates synthesis. So only one of the 2 strands of DNA called the template strand is transcribed. The strand of DNA that is not transcribed is called the coding strand. So the polymerase adds ribonucleotides to the growing 3' end of an RNA chain, And bacteria contain only one RNA polymerase enzyme, while eukaryotes have three different RNA polymerases. RNA polymerase 1 that synthesizes rRNA in the nucleolus, RNA polymerase 2 synthesizes mRNA, RNA polymerase 2 synthesizes tRNA. Now the promoter transcription starts at RNA polymerase binding sites called promoters on the DNA template strand. A promoter is a short sequence that is not itself transcribed by the polymerase but binds to it. So promoters differ widely in efficiency. Strong promoters cause frequent initiations of transcription as often as every 2 seconds in some bacteria. And weak promoters may transcribe only once every 10 minutes. So in bacteria, subunit of RNA polymerase called the sigma factor recognizes the -10 sequence in the promoter and the binds RNA polymerase there. So importantly, this subunit can detect the -10 sequence without unwinding the DNA double helix. So in eukaryotes, the -25 sequence plays a similar role in initiating transcription as it is the binding site for a key protein factor. Other eukaryotic factors then bind one after another, assembling a large and complicated transcription complex. So once bound to the promoter, RNA polymerase begins to unwind the DNA helix. Unlike DNA synthesis, a polymerase is not required. The region containing the RNA polymerase DNA and growing RNA transcript is called the transcription bubble because it contains a locally unwound bubble of DNA. Transcription bubble moves down the DNA at a constant rate, about 50 nucleotides per second, leaving the growing RNA strand protruding from the bubble. After the transcription bubble passes, the now transcribed DNA is now rewound as it leaves the bubble. So unlike DNA polymerase, RNA polymerase has no proofreading capability. Transcription thus produces many more copying errors and replication. So most genes are transcribed many times, so a few faulty copies are not harmful. At the end of a gene, our stop sequences that cause the formation of a phosphodiester bonds to seize the RNA polymerase to release the DNA and the DNA within the transcription bubble to rewind. The simplest stop signal is a series of GC base pairs followed by a series of AT base pairs. So the RNA transcript of this stop region forms a GC hairpin followed by 4 or more U-ribonucleotides. So how does this structure terminate transcription? The hairpin causes the RNA polymerase to pause immediately after the polymerase has synthesized it, placing the polymerase directly over the run of 4 uracils. Now the pairing of U with DNase, A is the weakest of the 4 hybrid base pairs and is not strong enough to hold the hybrid strands together during the log pause. So instead, the RNA strand dissociates from the DNA within the transcription bubble and transcription stops. A variety of protein factors aid hairpin loops in terminating transcription of particular genes. Now translation. In prokaryotes, translation begins when the initial portion of an mRNA molecule binds to an RNA molecule in a ribosome. So the mRNA lies on the ribosome in such a way that only one of its codons is exposed at the polypeptide making site at any time. So a tRNA molecule possessing the complementary 3 nucleotide sequence or anticodon binds to the exposed codon on the mRNA. So because this tRNA molecule carries a particular amino acid, that amino acid and no other is added to the polypeptide in that position. Now as the mRNA molecule moves to the ribosome, successive codons of the mRNA are exposed and a series of tRNA molecules bind one after another to the exposed codons. Each of these tRNA molecules carries an attached amino acid which it adds to the end of the growing polypeptide chain. And there are about 45 different kinds of tRNA molecules. Why are there 45 and not 64 tRNAs? How do particular amino acids become associated with particular tRNA molecules? The key translation step which pairs the 3 nucleotide sequences with appropriate amino acids carried out by a remarkable set of enzymes called activating enzymes. So activating enzymes called amino acid tRNA synthetases, one of which exists for each of the 20 amino acids. So therefore, these enzymes must correspond to specific anticodon sequences on a tRNA molecule as well as particular amino acids. So some activating enzymes correspond to only one anticodon and thus only one tRNA molecule. Others recognize 2, 3, 4 or 6 different tRNA molecules, each with a different anticodon but coding to the same amino acid. We have already talked the genetic code. There is no RNA with the anticodon complementary to three other 64 codons UAG, UGA, and UAA. These codons, the nonsense codons, serve as stop codons in the mRNA message, marking the end of a polypeptide. The start signal that marks the beginning of a polypeptide within an mRNA sequence is the codon AUG, which also codes for the amino acid methionine. Okay. So ribosome will usually use the first AUG that it encounters in the mRNA to signal the start of translation. Okay. So initiation in eukaryotes and prokaryotes is similar, although it differs in two important ways. First, in eukaryotes, initiating amino acid is methionine rather than N-formyl methionine. Second, the initiation complex is far more complicated than in bacteria, containing 9 or more protein factors, mainly consisting of several units. In elongation, when a tRNA molecule with the appropriate anticodon appears, proteins called elongation factors assist in binding it to the exposed mRNA codon at the A site. So when the second tRNA binds to the ribosome, it places its amino acid directly adjacent to the initial methionine, which is still attached to the tRNA molecule, so which in turn is still bound to the ribosome. The two amino acids undergo chemical reactions and catalyzed peptidal transferase, which releases the initial methionine from its tRNA and attaches it instead by a peptide bond to the second amino acid. Translocation: in a process called translocation, the ribosome now moves 3 more nucleotides along the mRNA molecule in the 5' to 3' direction. This movement relocates the initial tRNA to the E site and ejects it from the ribosome, repositions the growing polypeptide chain to the P site, and exposes the next codon on the mRNA at the A site. And termination elongation continues in this fashion until a chain terminating nonsense codon is exposed, for example UAA. Nonsense codons do not bind to tRNA, but they are recognized by release factors, proteins that release the newly made polypeptide from the ribosome. If we talk about the central dogma, we know that the flow of information in a cell starts at DNA, which replicates to form more DNA. Information is then transcribed into RNA, then it is transcribed into protein. The proteins do most of the work in the cell. Information does not flow in the other direction. This is a molecular version of the incorrectness of inheritance of acquired characteristics. Changes in proteins do not affect the DNA in a systematic manner. Okay. So we have talked about all of these previously, so I'm not going into much details. This question was more about genetic code. So we have discussed about the genetic code. You can go through the papers and the Watson and book of molecular biology. I think that will help you. These are chapter images taken from the book. So coming back to the question that we started with, bacterial mRNA codons are similar to other animal codons. This will be the answer. If anybody has any doubt, I'll wait for a few minutes, then ask the doubt, and then we'll move on to the next question for today. Okay, so no question. We'll move on to the next question for today. Telomeres come under the enzyme category A. Reverse transcriptase, B. Nuclease, C. DNA polymerase, and D. DNA ligase. Which one do you think is the correct answer? I'll wait for a few minutes. Anyone? Okay, the answer will be reverse transcriptase. I think we have discussed telomeres and telomeres in much detail in the last class. So if you remember, that's good. Otherwise, let me give you a little recap. So, what are telomeres? What are they? Why are they important? Telomere shortening and then replication problem. What is telomeres? What is the telomere hypothesis of aging, etc. So telomeres are basically ends of linear chromosomes. Okay, so we have the centromere in the middle and telomeres on both sides. It's a repetitive DNA sequence like TTA, GGG invertebrates. Specialized proteins, they form a capped end structure. So telomeres cap chromosome ends. If you see, these are how the telomeres look like, and you can see this is a fluorescent microscopy image. Why are telomeres important? Telomeres allow cells to distinguish chromosomes ends from broken DNA. Okay, like stop the cell cycle, repair or die. So homologous recombination error free but need nearby homologs, non-homologous anytime but error, right? So why telomere is important? Because it prevents chromosome fusions by NHEJ. What is NHEJ? Non-homologous end joining method. Telomeres also provide a means for counting cell division, right? So how do cells know how many divisions they have completed? This is a means of finding out that. So what is the end replication problem? DNA replication is bidirectional. Polymerase moves 5' to 3'. It requires a labile primer. So each round of DNA replication leaves about 50 to 200 base pairs DNA unreplicated at the 3' end. So telomere also provides a means for counting cell division, like telomere shorten with each cycle. Telomere shorten from 10 to 15 kb to 3 to 5 kb after 50-60 doublings. Cellular senescence is triggered when cells acquire one or a few critically short telomeres. Now, how do replicatively immortal cells avoid complete loss of telomeres? How do they solve basically the end replication problem? So, enzyme like reverse transcriptase with RNA and protein components like adds telomeric repeat DNA directly to 3' overhang, uses its own RNA as a template, and vertebrate repeat DNA on 3' end TTAGGG telomerase RNA template AAUCCC, right? So this telomerase is the key to replicative immortality. It overcomes telomere shortening and the replication problem. It's expressed by germ cells, early embryonic cells. It's not expressed by most somatic cells in human. May be expressed by some stem cells, but highly controlled. Expressed 80 to 90% of cancer cells. Remaining still need to overcome the replication problems, or do so by combination mechanisms. ALT is alternative lengthening of telomeres mechanism. So telomere length and cell division potential. If you see the y-axis is telomere length in humans and the number of doublings. This is how the telomere length and differs in all the different types of cells. For germ cells, telomere is positive. For normal somatic cells, telomere is negative. So it's going down with the number of doublings. The telomere length is going down. But if you have telomeres, it will reach a particular stage. However, cells that express telomeres still undergo senescence in response to DNA damage, oncogenes, etc. So, what are the biomedical uses? It expands cells for replacement therapies like burns, joint therapies, joint replacements, etc. And telomerase inhibitors to selectively kill cancer cells. So, the telomere shortened with each cell division. True. Short telomeres cause cell senescence, and senescent cells may contribute to aging. Again, true. So, telomere shortening causes aging, and telomeres will prevent aging. Is there a hypothesis? Is that true or false? That's basically the entire question. So, telomere length is not related to lifespan. Okay, telomeres contribute to aging only if senescent cells contribute to aging. So, telomeres protects against replicative senescence, but not senescence induced by other causes. So, in the summary, our telomeres are essential for chromosome stability. Telomere shortening occurs owing to the biochemistry of DNA replication. Short telomeres cause replicative senescence. Other senescence causes a telomere independent, and telomeres prevents telomere shortening and replicative senescence. And the telomere hypothesis of aging depends completely on the cellular senescence or hypothesis of aging. Okay. Now, telomeres are basically the structures at the ends of linear eukaryotic chromosomes. So, as I said, they consist of a series of unique DNA sequences and specialized proteins that bind to them. And telomeres adds a unique sequence on the ends of eukaryotic chromosomes. So, discovered in tetrahymena micronucleus, and they have the sequence TTGGG in humans. And okay, so this is the T loop. The telomeres needed at the ends of chromosomes for several reasons. Telomeres prevent the chromosome ends from being ligated together. Telomeres prevent the ends of chromosomes from being attacked by exonucleases. The loop structure explains the reasoning for both. Telomeres attach chromosomes to the NE nuclear envelope lamina. Telomeres begin pairing or synapses of homologous chromosomes during meiosis. Telomeres can compensate for incomplete replication of linear chromosomal ends. So this is telomere action. Okay, so if you see this molecular remainder of telomere is RNA, telomere is protein fingers, and region of telomere is RNA uses template. Palm or active site of telomere is protein. Newly synthesized telomere is DNA. Okay. Now, if you talk about centromeres, molecular biology, we have discussed all of these, so I'm not going in detail. So, coming back to the question that we started with for today, telomeres comes under the enzyme category of reverse transcriptase. Okay. If anybody has any doubt in this question, we can stop here for a few minutes and then we'll move on to the next question for today. So we'll move on to the next question for today. Wait a minute. 1 second. So there's 1 second. Yeah. So next question for today is cancer risk is high when A. Telomere is inactive, telomere is short, P53 is active, telomere is active. I'll wait for a few minutes. If you know the answer, you can tell me. Then we'll move on to the next question for today. So the answer will be telomerase is active. Okay, right? Why is it that cancer risk is high when telomerase is active? So if we start talking about this topic, we have talked about telomerase and telomerase, right? So while telomerase activation is a hallmark of many cancers, allowing cells to divide indefinitely, it's not solely the cause of increased cancer risk, right? So telomere shortening and dysfunction also contribute to cancer development. So as you know, the telomerase is an enzyme that maintains telomere length, the protective caps at the ends of chromosomes. In most normal somatic cells, telomerase activity is low and telomeres gradually shorten with each cell division, eventually leading to cell senescence or death. However, in the vast majority of cancer cells, telomerase activity is reactivated, allowing them to maintain telomere length and divide indefinitely, a key characteristic of cancer. So this telomerase reactivation along with other factors contributes to uncontrolled cell growth and tumor development, right? Telomere shortening can also contribute to cancer development. While telomeres become critically short, cells can enter a state of crisis, leading to genomic instability and increased cancer risk. And individuals with shorter telomeres may have a higher risk for certain cancers. Now, what is the dual role of telomeres? Telomeres play a complex role in cancer development, with both telomere shortening and telomerase activation contributing to the process. Telomere shortening can trigger cell senescence or apoptosis, which can be a protective mechanism against cancer. However, telomere shortening can also lead to genomic instability and increased cancer risk. Now, how do we target telomerase for cancer therapy? So, due to the high levels of telomerase activity in cancer cells, telomerase has become an attractive target for cancer therapies. So researchers are exploring ways to inhibit telomerase activity in cancer cells, potentially leading to the development of new cancer treatments. So while telomerase activation is a hallmark of cancer, telomere length itself can also be a risk factor. So both short and long telomeres have been associated with increased risk cancer. So individuals with very short or very long telomeres may be at a higher risk of developing cancer. Right? So are telomeres the key to aging and cancer? So inside the nucleus of a cell, our genes are arranged along twisted double-stranded molecules of DNA called chromosomes. So at the ends of our chromosomes are stretches of DNA called telomeres which protect our genetic data, make it possible for cells to divide, and hold some secrets to how we age and get cancer. So telomeres have been compared with the plastic tips on shoelaces because they keep chromosome ends from fraying and sticking to each other, right? So which would destroy or scramble an organism's genetic information. So yet each time a cell divides, the telomeres get shorter. So when they get too short, the cell can no longer divide, right? It becomes inactive or senescent or it dies. So the shortening process is associated with aging, cancer, and a higher risk of death. So telomeres have also been compared with the bomb fuse. Like if you see the image here, fluorescent stained chromosomes on a microscope slide. So telomeres are the yellow stained, right? It's a fluorescence microscopy image. Now what are telomeres? We have discussed about it already, but again I will short recap. When DNA replicates during cell division, a short piece of RNA called a primer helps to get it started. So once a primer attaches, cellular machinery can copy the DNA strand. Now since the primer does not attach to the very end of the DNA strand, the copy is missing a section of DNA. So the next time the cell divides, the copy DNA loses more of the end section. So as cell division continues, the end section of each new DNA strand gets shorter and shorter. See, in humans, about 50 to 250 base pairs are lost per cell division. So telomeres are repeated sequences on the ends of DNA strands. They help protect the DNA strand from getting shorter during cell division. Now in germline cells, an enzyme called telomerase is responsible for adding more repeat sequences to the end of the DNA, thus making them immortal. And in somatic cells, the telomerase enzyme functions at a much lower level, making these cells mortal. So when the telomeres in a somatic cell shorten to a critical level, that cell no longer divides. So this phenomenon contributes to some of the changes we see in aging. So like the rest of a chromosome, including its genes, telomeres are sequences of DNA, chains of chemical code. Like all DNA, they are made of 4 nucleic acid bases: G for guanine, A for adenine, T for thymine, and C for cytosine. Telomeres are made of repeating sequences of TTA-GGG on one strand paired with AATCCC on the other strand. Thus, one section of telomere is repeat made of 6 base pairs. So in WBCs, the length of telomeres range from 8,000 base pairs in newborns to 3,000 base pairs in adults, and as well as 1,500 in elderly people. Right? So each time it divides, an average cell loses 30 to 200 base pairs from the ends of its telomeres. And cells normally can divide only about 50 to 70 times, with telomeres getting progressively shorter until the cells become senescent or die. So telomeres do not shorten tissues where cells do not continually divide, such as heart muscle. So why do centromere chromosomes have telomeres? Again, without telomeres, the main part of the chromosome, the part with genes essential for life, would get shorter each time a cell divides. So telomeres allow cells to divide without losing genes. Cell division is necessary for growing new skin, blood, bone, and other cells. So without telomeres, chromosome ends could fuse together and corrupt the cell's genetic blueprint, possibly causing malfunction, cancer, or even cell death. So because broken DNA is dangerous, a cell has the ability to sense and repair chromosome damage. Without telomeres, the ends of chromosomes would look like broken DNA, and the cell would try to fix something that wasn't broken. So that would also make them stop dividing and eventually die. Now, why do telomeres get shorter each time a cell divides? So before a cell can divide, it makes copies of its chromosomes so that both new cells will have identical genetic material. So to be copied, a chromosome's 2 DNA strands must unwind and separate. An enzyme then reads the existing strands to build new strands. So it begins the process with the help of short pieces of RNA. When each new matching strand is complete, it's a bit shorter than the original strand because of the room needed at the end for this small piece of RNA. So it's like someone who paints himself in the corner and cannot paint the corner, right? So enzyme named telomere adds bases to the ends of telomeres. In young cells, telomeres keeps telomeres from wearing down too much. But as cells divide repeatedly, there is not enough telomeres. So the telomeres grow shorter and the cells age. Now, telomeres remains active in sperms and eggs, which are passed from one generation to the next. If reproductive cells did not have telomeres to maintain the length of the telomeres, any organism with such cells would soon go extinct. Now, as a cell begins to become cancerous, it divides more often, and its telomeres become very short. So, if its telomeres get too short, the cells may die. So, oftentimes these cells escape death by making more telomerase enzyme, which prevents the telomeres from getting even shorter. So, many cancers have shortened telomeres, including pancreatic, bone, prostate, bladder, lung, kidney, head, and neck. And measuring telomeres may be a way to detect cancer. And if scientists can learn how to stop telomeres, they might be able to fight cancer by making cancer cells age and die. So in one experiment, researchers blocked telomerase activity in human breast and prostate cancer cells growing in the lab, prompting the tumor cells to die. But there are risks. Blocking telomeres could impair fertility, wound healing, and production of blood cells and immune system cells. Geneticist Richard Cawthon and colleagues at the University of Utah found shorter telomeres are associated with shorter lives. So among people older than 60, those with shorter telomeres were 3 times more likely to die from heart disease and 8 times more likely to die from infectious diseases. So while telomere shortening has been linked to the aging process, it is not yet known whether shorter telomeres are just a sign of aging, like gray hair, or actually contribute to aging. So if telomeres makes cancer cells immortal, could it prevent normal cells from aging? Could we extend lifespan by preserving or restoring the length of telomeres with telomeres? If so, would that increase our risk of getting cancer? Scientists are not exactly sure, but they have been able to use telomeres in the lab to keep human cells dividing far beyond their normal limit, and the cells do not become cancerous. So if we use telomeres to immortalize human cells, we may be able to mass produce cells for transplantation, including insulin producing cells, to cure diabetes, muscle cells for treating muscular dystrophy, cartilage cells for certain kinds of arthritis, and skin cells for healing severe burns and wounds. So, an unlimited supply of normal human cells grown in the lab would also help efforts to test new drugs and gene therapies, right? So, how big is the role of telomeres in aging? So, what are some of the factors in aging? Telomere shortening, chronological age, oxidative stress like oxidants damage DNA, proteins, and lipids, and glycation. Glucose, sugar, it binds to and inhibit DNA, protein, and lipids. Some long-lived species like humans have telomeres that are much shorter than species like mice, which live only a few years. Nobody knows why, but it's evident that telomeres alone do not dictate lifespan. So, Cawthorn's study found that when people are divided into two groups based on telomere length, the half with longer telomere lives an average of 5 years longer than those with shorter telomeres. So, this study suggests that lifespan could be increased 5 years by increasing the length of telomeres in people with shorter ones. So, people with longer telomeres still experience telomere shortening as they age. How many years might be added to a lifespan by completely stopping telomere shortening? Cawthorn believes maybe 10 years and even perhaps 30 years. So after age 60, the risk of death doubles every 8 years. So a 68-year-old has twice the chance of dying within a year compared with a 60-year-old. So Cawthorn's study found that differences in telomere length accounted for only 4% of that difference. And while intuition tells us older people have a higher risk of death, only 6% is due purely to chronological age. When telomere length, chronological age, and gender are combined, women live longer than men. These factors account for 37% of the variation in the risk of dying over age 60. So, what causes the other 63%? So, a major cause of aging is oxidative stress. It is the damage to DNA, proteins, and lipids caused by oxidants, which are highly reactive substances containing oxygen. And these oxidants are produced normally when we breathe and also result from inflammation, infection, and consumption of alcohol and cigarettes. So, in one study, scientists exposed worms to two substances that neutralize oxidants, and the worm's lifespan increased on average 44%. So, another factor in aging is glycation. It happens when glucose, the main sugar we use as energy, binds to some of our DNA, proteins, and lipids, leaving them unable to do their jobs. The problem becomes worse as we get older, causing body tissues to malfunction, resulting in disease and death. Right? So, glycation may explain why studies in laboratory animals indicate that restricting calorie intake extends lifespan. Most likely, oxidative stress, glycation, telomere shortening, and chronological age, along with various genes, all work together to cause aging. Now, telomeres and other diseases. People with a disease named dyskeratosis congenita have telomeres that get short much more quickly than normal, and these people endure premature aging and death, so they face a higher risk of life-threatening infections, leukemia and other blood cancers, intestinal disorders, cirrhosis of the liver, and pulmonary fibrosis, a deadly stiffening of lung tissue. They also are more likely to endure gray hair, balding, poor wound healing, spots on the skin, intestinal disorders, softening of the bones, and learning disabilities. So the implication is that telomeres may play a role in all these conditions because they all involve tissues in which cells divide often. So there also is some evidence linking shortened telomeres to Alzheimer's disease, hardening of the arteries, high blood pressure, and type 2 diabetes. So what are the prospects for human immortality? Human lifespan has increased considerably since the 1600s, when the average lifespan was 30 years. By 2012, the average US life expectancy was nearly 79. So reasons for the increase include stress and other sanitation measures, obviously, antibiotics, clean water, obviously, vaccines and other medical efforts, medical science advancement to prevent children and babies from dying, improved diets and better health care. Some scientists predict average life expectancy will continue to increase, although many doubt the average will be ever much be higher than 90. But a few say vastly longer lifespans can be possible. So, Cawthorn says that if all processes of aging could be eliminated and oxidative stress damage could be repaired, one estimate is people could live even 1,000 years. Okay. So, We know about cancer and what happens during cancer, protein cell control, cell cycle, all of these we know. Now cells anchor to dish surface and divide. When cells have formed a complete single layer, they stop dividing. Density independent inhibition. If some cells are scraped away, the remaining cells divide to fill the dish with a single layer and then stop. Density dependent inhibition. After forming a cell, provide an additional supply of glucosidase for the cell division. Okay. So we'll not go into the traits of cancer. These are too much details into cancer. We have already discussed about telomeres, what the question was all about. Right. Wait 1 second. So the question that we started with was cancer risk is high when telomere is active. So that is what it's all about. Okay. Does anybody else have any questions regarding this uh, particular question? If not, then we can stop the session for today and we'll meet again next week. Okay, if nobody has any doubt, we can stop the session here for today. So if nobody has any doubt, we can stop the session here for today. And we'll meet again next week, same time, same day, same time, Monday next week. Okay? So we'll stop the session. to project